Welcome back, Alan Lockwood.
Well, thank you very much, Marjorie, and thank you all for coming out on our last unmasked night in the auditorium, I think. Hopefully that won't last for too long. So as you see, the title of my presentation tonight is America's Threatened Forests and Restoring the American Chestnut. This story began a little over 2 years ago when I happened to see, and I'm not quite sure how I saw it, but I did notice that the environmental sciences and forestry college at the Syracuse University campus of SUNY, State University of New York, had filed a petition with various federal agencies to introduce a blight-tolerant form of the American chestnut, and they were looking for comments to be posted on in the federal docket. So David Benzing and I wrote some comments, sent them in. I sent a copy to the PI who is heading the ESF initiative, and he encouraged us to apply for partner status in the plan to distribute the now known as the Darling 58 American chestnut at some time in the future, pending federal approvals. And I'm going to talk a bit about that, and I want to put it in the context of why this is an important initiative and what's happening to America's forests that makes this important. Now, when I first began thinking about this, of course, it was Thanksgiving time and was reminded of the story, true or not, that pilgrims and native peoples gathered to celebrate the first Thanksgiving. Accounts that I've read since then made it sound like the pilgrims were making a ruckus, firing off cannons and guns, and the Indians came over to see what was going on, and one thing led to another, and the Native American people started eating, and the Americans stood there and watched. But a woman who's Ramona Peters, who's the historic preservation officer for the Mashpee Wampanoag tribe, believes that the food that was consumed by the native peoples included Concord grapes, native nuts, including walnuts, and chestnuts. So that led me to further thoughts about, the chestnut is now functionally extinct, and we're standing on lands that used to belong to native people. So I thought it would be appropriate to just think for a moment and remember that we were not the first ones here, and we really do owe a debt to the people who came before us. So in my talk tonight, I want to present an overview of the threats to forests in the United States. These are due primarily to pests, disease, and climate change. And I want to extrapolate to some considerations about how this is consonant with what's expected to happen to agricultural commodities. I want to focus on the American chestnut and chestnut blight, which has basically destroyed the once iconic tree from eastern forests and rendered it functionally extinct. And the attempts to bring back the American chestnut by two different strategies, back-cross breeding and development of the Darling 58 tree. And as I will tell you a number of times, if fortune smiles on all of us, we will get Darling 58 trees next summer. And finally, I want to conclude with what the role of the John Bartram Arboretum is expected to be in the restoration of the American chestnut. See, I managed to screw up already here. So this story begins with the efforts of the United Nations Decade for the Ecosystem, and I think we heard a mention of this earlier this week. And as a part of that decade for the ecosystem efforts, there's been an effort to catalog all of the trees that are in existence in the entire globe. And the Global Tree Assessment has identified something like 60,000 known species worldwide. And they've created a checklist of tree species that are native to the United States and found that there's something like 881 species from 269 genera that are found in the country, and the most common genera in the U.S. are Quercus, the oaks, and Crataegus, the hawthorns, and related plants. I'll tell you just a bit more about that. I hope you can see some of these numbers at least. This table lists some of the results of the survey of U.S. species that is in descending order of the number of species in the genus. And for oaks, there are 85 different species of oaks in the United States, of which 42 are so-called endemic species. And endemic species are those that occupy a limited geographical area. And for the, you see here the Quercus or the oaks, 85 species, hawthorns, 84, and then we come down to the yuccas, which are 11 different species in that genus, with 2 that are in the endemic category. Now we have quite a few oaks on the campus here in the Bartram Arboretum, and this is shown by this screenshot from the Arborscope utility, which you can access through the website. If you go to the home page of the website and click on maps and walks, etc., you will then see a drop-down menu and go through to the Arborscope utility, which is a little bit kludgy. At least on my computer, it takes a little while for it to fire up. As you can see, there are quite a few oaks on campus, including a couple that we're probably not too pleased to have in our collection because they're categorized as invasive. And in the Ohio Department of Natural Resources, they encourage you to not plant these in the state. But we've got 2 of them at least, maybe more. And here are the hawthorns, the smaller number, perhaps, but there are probably, again, many more that are not shown on this because some of them are likely to be shrubs and don't show up in the official catalog of the Arboretum collection. Now, the native tree species in the United States are distributed mainly with the highest density in the southeastern portion of the United States, Texas and Florida having the largest numbers with 300 plus different species. There's some 80-some species in the state of Ohio. So that if we're looking ahead to a time when someone might want to apply for level 3 status for the Arboretum, you have to have at least 500 different species of trees in the collection in order to get to that level. We have about 120, 125 different species. And so that would be about 2/3 of all the species in the United States represented in our collection. It's not too likely to happen. Plus, we would also need a substantial increase in funded positions for conduct of educational programs and scientific work and stuff like that. So we will be content with our level 2 status for quite some time. Now, the risk for extinction is found to range between 11 and 16% of all of the tree species in the country. And the major threats are due to invasive and problematic pests, diseases such as the emerald ash borer, which is a pest, and chestnut blight, which is a disease. Climate change and severe weather also play a role, and degradation of habitats, usually due to human activities, need to be contended with. And biological resource use, such as harvesting for logging to make paper, lumber, and so on and so forth. So a lot of the threats that are posed to U.S. tree species are due to humans. just to look at this a little bit more closely to drill down on this, the number of threatened species is listed here in this column in descending order. The hawthorns have 29 of the 84 species that are thought to be threatened. Oaks, 17 of the 85. If you look at the ash genus, that's almost half of all of the ashes fall into this threatened category. There aren't as many genus in the United States of ashes, but those that we do have are under quite an intense pressure. And to look at this a little bit more closely in the case of oaks, which is the most common genus, Quercus, the most common genus in the United States. Oops. Probably those of you who are in the front might be able to see this. The dark red are species of conservation concern, and across the x-axis is the number of species. So it's somewhere between 5 and 10, probably closer to 8.5 or so, of all oak species are threatened by habitat shifting and alteration. Fire and fire suppression is next. Livestock farming, problematic species introduced of unknown origin, about 5%, and it goes down here. Looking at this recreational activities, almost 10%, close to 9 or so. So that human activities are playing a big role in putting pressure on oak species. And it's thought that oaks were the trees that moved in and filled the vacant spots that were left when chestnuts died. At one time, it was thought that there were between 3.5 and 4 billion American chestnuts in the eastern forests. So here's an example of one of the pests that's causing lots of trouble, including here on our campus and in the trees and the forests around us. The emerald ash borer. This is not life-size, but you can see here's where the larvae have been digging around in the cambium of the tree, and they effectively girdle the tree so that nutrients are unable to flow up and down the trunk of the tree and nourish the tree, and the tree dies. So there are a number of ash trees that have been cut down that used to be in our collection. There's one as you move away from Kendall and go across the Troll Bridge, there's a cluster of suckers that have sprouted up out of a dead ash tree stump, but those two will soon be affected by the emerald ash borer. Another pest is the mountain pine beetle, and here's a forest of lodgepole pines in British Columbia that have been killed by the action of the activity of this mountain pine beetle. And this dead trees combined with the drought that is at millennial degrees of severity has resulted in the drying out of a lot of the forests in the western parts of the United States and Canada, and it is one of the factors that's making wildfires so dangerous and so prevalent in that part of the country. Oops. Now, when a species like a chestnut goes extinct, trees, other species that depend on the existence of the chestnut also are likely to go extinct. And here's a quotation from David Wagner, who is quoted in this news report, who said that the American chestnut extinction correlates to the greatest invertebrate extinctions on Earth in the modern era. That there are only 61 invertebrate extinctions in the modern era, 41 in North America, and of these 5 are directly related to the loss of the chestnut. And other authors have written in Science that perhaps there are over 6,000 species that are currently not endangered, but would go co-extinct if the counterpart tree or plant that they depend on for their existence was to go extinct. So it's a sort of a domino theory. You wipe out a keystone species and other plants and animals that depend on that species in order to exist, face extinction too. Now the American chestnut grew to enormous size, as you can see in these photographs of these three people sitting in the notch of this tree that's about to be felled. A photograph from the American Chestnut Foundation, and another photograph from the Annandale, Virginia Chamber of Commerce shows that this chestnut tree was about 18 feet in diameter. So it's sort of reminiscent for those of us who have been out on the West Coast and seen the coastal redwoods and the giant sequoias. You know, they're almost of the magnitude of some of those gigantic trees. So this was truly a giant of the eastern forest, and they're gone. Now, the American chestnut, for those of you who got here early enough to hear the beginning of Frank Sinatra's crooning, that song starts off, chestnuts roasting on an open fire. It's like, just couldn't resist playing that while people were coming in. But here's an etching or an engraving by Winslow Homer showing a family out gathering chestnuts, and these were an important part of the food supply of early American settlers, native people. Lots of animals and birds depended on them, and when the chestnut disappeared, those species were all put under stress. And here's another American chestnut. This one happens to be dead in a photograph that was taken in the Smokies in 1920. Now, luckily, there are still a few genuine American chestnuts that are still living. The ones that are shown in this photograph are not living. One of them was thought to have been planted in 1885 and tended to by the sixth great-grandson of Daniel Boone. Now, I don't know who figured that out. Somebody who had too much time on their hands. But there are a few places where there are isolated populations of American chestnuts that still survive. But those are fewer and far between. Now, the organism that causes chestnut blight, Cryophonectra parasitica, pretty apt species name, was introduced into the United States and Europe in the early part of the 20th century, probably from the Far East. It spread rapidly and killed between 3 and 4 billion trees in eastern forests of the United States. This same fungus also infects oaks, maples, staghorns, sumacs, shagbark hickories, and others, but it doesn't kill them. They're tolerant of the infection. The infection produces initially an orange bark with cankers that girdle and kill the tree. And this is a picture of a canker that I took at the Environmental Sciences and Forestry campus this past summer when Ann and I stopped in and visited them. The first signs of the blight are an orange-brown discoloration of the bark. And as the fungus grows, it develops a fan of the mycelia that spread throughout the cambium of the tree and secrete toxic compounds, most notably oxalic acid, that lowers the pH of the tissue to the point where it can't survive, causing death of the cells where the fan of the fungus has spread. And again, like the emerald ash borer, it girdles the tree once the canker goes all the way around the tree and causes death of the tree. And that gave rise to the hypothesis that I'll come back to, that will some strategy for dealing with the oxalic acid and removing it be effective in making the American chestnut tolerant to the blight, and I'll come back to that point in some detail. Here's a couple of micrographs of the chestnut blight fungus. As I said before, it infects through a flaw in the bark, And through that defect, it reaches the inner bark and the cambium, where it sets up housekeeping and secretes oxalic acid as the mycelial fan spreads. And then after the fungus spends a winter, the first rains that fall and moisten the bark of the tree trigger the release of fruiting bodies from the fungus, and that is how it spreads. So the released sexual spores are then spread by air or by birds or insects or other vectors infecting other trees. And ironically, it's probable that some of the insects that are now extinct as a result of the loss of the American chestnut were the same ones that landed on the tree and had some of these spores attach themselves to the insect when it flew off to another tree. And this is what that looks like. It sends out these little things that look like spaghetti, and then the sexual spores erupt from that and spread to adjacent trees. So there have been a couple of strategies that have been developed to try to preserve the American chestnut, and much to my surprise, I read in one of the sources that more money has been spent on trying to restore the American chestnut than has been given to support giant panda populations. So you see these things from World Wildlife Foundation with pictures of pandas on them, urging you to do a subtotal wallotectomy and give them some of your money. The American Chestnut Foundation and the research efforts that have gone into trying to preserve this iconic species has accounted for an enormous amount of spending. The two strategies that have been utilized the most are controlled back crosses, which is a strategy for controlled breeding of native American chestnuts with Chinese chestnuts. And I'll talk about that in some detail in a moment. And then also the strategy for using genetic engineering techniques that starts with an understanding of the pathophysiology of chestnut blight, and then engineering a plant that's able to tolerate the blight. This is really close to my heart, because this is how we're taught in medicine how to approach diseases. You start with an understanding of the physiology and the pathophysiology, and then you devise a treatment strategy that interrupts that disease process in some way that is tolerable by humans, and get rid of the problem. Like taking penicillin for pneumococcal pneumonia. Now, in order to be successful at either of these strategies, you have to be able to control the pollination of chestnut trees. And chestnuts have both male and female parts on them, but they're not able to self-fertilize, so that they rely on cross-fertilization between trees, which creates an opportunity to place bags, as you see in these trees here on the right-hand side of the screen, that isolate the female parts of the tree so that pollens from unwanted or unknown chestnuts don't fertilize the female part of the tree. And here you can see immature catkins, which are the name of the male parts of the tree, and this shows a more mature catkins, and this is where the pollen comes from, travels to an adjacent tree and fertilizes a female part. So it although they have both sexes, it takes two trees to do the job. And this is how backcross breeding works. It's a complicated process and relies on controlled fertilization using those kinds of bags there that you saw on the trees in the prior slide there. So you start with a Chinese chestnut and a native American chestnut, and you create a cross between the two of them. So the progeny from that first cross have half of their genome from the Chinese chestnut and half from the American chestnut. Then you take the progeny from that look to see which ones do the best in terms of blight resistance, and then breed them again, the product of that, with another American chestnut, the real McCoy. And then you do the same thing again. So now you're down to a quarter of the genome being due to genes from the from the Chinese chestnut, and 3/4 from the American chestnut. Do that one more time, and you have 1/16 of the genome being from the Chinese chestnut, and the rest, the other 3/4, the 16th due to 16th due to the Chinese chestnut, and then the 15/16 due to the American chestnut. And then you start breeding these trees that are 1/16 Chinese chestnut and 15/16 with each other, and by the time you do that 3 times, you get what is referred to as backcross 3 of these trees here, the 3rd generation. So you go through 6 different generations of trees before you get something that is most reliably going to be resistant to the blight. But meantime, you've had to gain control of all of the sources of pollen and do the controlled breeding. Takes about 5 years between generations of trees. And the literature says that you go through about 20,000 trees before you wind up with a tree that you're pretty confident is going to do the job. But even then, these trees are not completely resistant to the blight. They will get affected and may die. Now we have 3 of these hybrid trees of unknown genetic makeup in our collection on Wildflower Hill, and right now they're all in pretty good shape. So maybe they're these so-called BC3 F3 sourced trees. So this is a labor-intensive and expensive way of doing it, but it's attracted a lot of attention, and a lot of effort has gone into that, both from the Department of Agriculture and other chestnut breeding. Now I'm going to tell you a bit about the genetic engineering that goes along with insertion of the oxalate oxidase gene into the DNA of the genuine American chestnut. So it starts with isolating this oxalic acid oxalate gene and exposing a bacterium called agrobacterium, which is a bacterium that's used quite a lot in plant genetics. I'll have to take their word for it. I'm not a plant geneticist, but that's what it says in the literature. So this fragment of the gene containing oxalic acid oxidase gets incorporated into the genome of this bacterium that then injects this into the host, which in this case is an embryo from a genuine American chestnut tree, not a hybrid or anything. This is the real McCoy. So the gene goes out through a pore into the cytoplasm of the embryo, travels around with an escort, goes through pores into the nucleus of the embryo, releases the gene fragment that then gets incorporated into the genome of the American chestnut embryo. Now, in some instances, they put a marker gene in there along with oxalic acid oxidase. And in some of the literature that you may, if you really get into the weeds there, they put a gene in there that causes the plant to glow in the dark if you shine ultraviolet light on it. So it's easy to tell which trees, which embryos have been satisfactorily infected and tagged with the oxalic acid oxidase. Now, after this process here that you see on the screen is done, you want to get rid of the bacteria, and they do that by exposing it to a cocktail of 3 very potent antibiotics, washing the antibiotics off. So now the bacterium is gone. There's a tiny bit maybe of the antibiotics that are left, but by the time you take a couple cell embryo and grow it into a giant tree and dilute any antibiotic that might be there, you almost certainly would not be able to detect that. So that's really not a source of any concern by anyone. So this is what the Darling 58 blight-tolerant chestnut looks like. This is on the left-hand side, and panel A is the real McCoy American chestnut that's been deliberately incised and infected with the blight. And you can see the beginning of the canker here. And over here is a Darling 58 that was treated in an identical fashion at the same time. And there is essentially no blight infection. There's no canker being formed there. And again, these were pictures that I took of trees when Ann and I visited the ESF site in Syracuse last summer. I'd like to shift gears a little bit and talk about some of the other threats to trees in American forests. And one of these is climate change. And I'm not going to show you a picture that shows land surface temperatures as a function of time. Probably everyone in the room knows that it's hotter now than it used to be, and that there's no good end to that in sight. But if you look through the United States Department of Agriculture at their climate change tree atlas, you can pick a tree, and in this case I chose the sugar maple since it's sort of a, who doesn't like to put maple syrup on their pancakes in the morning? There are some people, I guess. So this is the current range of the sugar maple in the United States. It's a bit like the American chestnut, but not completely. And this is what is expected to happen by the end of the century if an emissions strategy goes ahead unabated. Emissions continue as they are. So this where did my pointer go here? This area here is all habitat quality low and colonization likelihood null. So basically chestnuts are going, sugar maples are likely to disappear from all of this area and retreat up into the northern parts of Maine and Ontario and other parts. So trees can't pick up and move like people can. So that this is going to be a real problem for not just sugar maples, but other species that are sensitive to changes in temperature and other factors that are associated with climate change. Now, trees are not the only thing that are affected by temperature. This is a little bit off the mark, but very much related, in that this shows the effect of temperature on the yield of corn. Don, pay close attention. Don Reeves, who does farming in Nebraska. As the temperature goes up and reaches between 25 and 30 degrees Celsius, the yield of corn drops dramatically. And in the business as usual scenario for emissions, by the end of the century, it's expected that the yields of corn will fall to by about 80%, only 20% of what we get now. So something has to be done if we're going to, we've had 8 billion people on the earth. Large numbers of people are going hungry now that didn't used to be hungry because of the interruptions in the flow of crops that are coming from Ukraine and Russia because of the war. If you cut that down to 80% of by 80% of yields now, we're going to be in a real pickle by the end of or our grandchildren will. And the same thing is happening to other agricultural commodities. And these are data that were acquired between 1980 and 2008. And the yield of corn is down somewhere between 10 and 15% in Brazil, a little bit less severely affected in China. And in Russia, the yield of wheat is down to by almost 15%. So yields of agricultural commodities are already falling because of changes in weather and changes in temperature. And things are only going to get worse as time goes by. Now, I mentioned before, perhaps not very clearly, but I will now, that you just can't go out there and develop a new plant like the Darling 58 American chestnut that's genetically modified. You have to jump through a whole bunch of hoops in order to gain permissions from the federal government in order to be able to plant and release that. You can grow them under controlled circumstances, and that is exactly what's happening at the ESF orchards in Syracuse, near Syracuse. So federal rulemaking that is required in order to be able to get approval for release of these begins by Congress giving the executive branch the authority to make regulations that that affect various things. So it may be a decision by the agency so that the regulations that control national ambient air quality standards, such as particulates and ozone, oxides of sulfur and things like that, are done by agency decision making. Or someone can petition the agency, and that's what's happened in the case of the ESF effort. They have petitioned the federal government for permission to release Darling 58 American chestnuts into the environment without any controls on them. So that begins by placing a notice in the Federal Register, and then the agency science groups begin an evaluation and come up with an opinion. And meanwhile, the petition is open for public comment. And if you're having trouble getting asleep at night, you can just pick up the Federal Register and start reading it. So it's endless, it's huge, and it's pretty dry reading. But you know, there are people who get paid for paying attention to this kind of stuff. So after public comment, then there may be an extended period of public comment if there's enough interest or in doing so by various agencies. And in some cases, tens of thousands of comments are posted on the docket with regard to a proposed rule. Then the petitioner and the agency will, the agency will come up with an opinion, and then they will take into account the comments that have been received and come up with a summation of that. And then the petitioner likely goes back and modifies their petition to the agency to take into account the comments that have been posted in the docket. That may require doing additional experiments or clarifications or other reviews of the literature, and then they come back and repost their petition. And then there's another period of commenting that goes on. And this is where the process for gaining approval by ESF in order to be able to release the Darling 58 American chestnut is. So it's in a second stage of commenting, and we have prepared comments that will be submitted to the Federal Register docket in a few days in support of the ESF petition. So the agencies that have jurisdiction over the Darling 58 are the United States Department of Agriculture. It's a plant, and it's going to be grown and released. So the Department of Agriculture has control over plants. The Food and Drug Administration has authority because people are going to eat the nuts that come from the American chestnut. And then partly as a measure of goodwill, the people at ESF said, look, Environmental Protection Agency ought to have a say in this as well. So there are 3 different federal agencies that have jurisdiction over the release of the petition put forth by ESF scientists. And then there will be a final decision by the agencies, and this will be a combined effort on the part of the 3 agencies. And it'll be either thumbs up or thumbs down. If it's thumbs down, there'll be another period of revising the petition and more public comment. Hopefully that will not occur. Of course, once a rule has been posted, then it's subject to challenge in the courts, and Lord only knows what's going to happen there. Now, there are, in the comments and also in the petition itself, the scientists at ESF were very cognizant of safety concerns that are involved here. So just to review here, it has the oxalic acid oxidase enzyme here that converts oxalate with oxygen and hydrogen ion to carbon dioxide and hydrogen peroxide. Hydrogen peroxide fizzes off and causes no harm. In fact, there's some data that suggest that it may harden the wall of any canker that's being produced. But this same gene is found in a whole host of agricultural commodities, and there may not be anyone in the audience tonight that didn't eat something that was on this list here. Wheat, peanuts, wheat of course, because that's where the gene comes from, but it's in peanuts, oats, beets, tea, strawberries, barley, cacao. Thank goodness we can't have it. Chocolate would be on the bad guy list. Corn, bananas, rice, peaches and apricots, and probably other stone fruit as well. Spinach, palms, lots of agricultural commodities that are consumed safely by humans and animals with no ill effect. This gene is completely separate from the gene that produces gluten, so people who are gluten intolerant can cross that worry off of their worry list. So, so this is a highly distributed gene that's very safe, and feeding studies that have been done with oxalic acid oxidase have all been fine. Now, the nutritional content of the nuts that are produced by Darling 58s has also been analyzed by an independent laboratory. Wasn't in the laboratory of the people at ESF. They send it off to a certified laboratory, and then the certified laboratory sends the result back. And this is a summation of a very extensive list of nutrients, and this is more akin to what you would see on the label of something that you might pick up in the supermarket. How many calories are there in a serving? How much fat is there? Those of you who are watching your sodium, good news for you here. There is none in the nuts of chestnuts. There's carbohydrate and some protein. And if you ate 15 of the Darling 58 nuts, you get almost half of your vitamin C ration or recommended daily intake of that vitamin. So nutritionally, they're very similar to the chestnuts that were being gathered in that figure that I showed earlier in the talk. Now, another source of concern that was expressed by some commenters was the potential to exacerbate seasonal rhinitis like hay fever and asthma and so forth. But this is almost certain to be a very de minimis concern. Darling 58s do produce pollen, but it's likely to be a minor contributor to the total exposure that we have to pollen. First of all, the number of Darling 58 trees is going to be pretty small. If we're lucky, we'll get between 2 and 5 of them, and they'll be put up on Wildflower Hill, and I'll show you where in a few minutes. Now, we have at least 1,000 trees on the campus of Kendall, and if you look at the number of trees in Oberlin, there's thousands more. So 2 to 5 trees out of the 1,000 that are around us. Even though they do produce pollen, it's going to be diluted by the pollen that's being produced by other trees. Now, like many other things, there have been surveys about pollen production in the United States. And in the last couple of decades, there's been a 42.6% increase in the average peak pollen concentration and a 46% increase in the average annual daily pollen exposure. So there are stations all over the United States, none in Ohio. There's one in Erie, Pennsylvania, where they measure this stuff. Now, also the pollen from chestnuts is pretty heavy and doesn't travel very far. And in the report in the Chestnut Pollinators Guide, it says that trees that are 100 feet apart are expected to have limited pollination success. And if they're 1,000 feet apart, they're reproductively isolated. So put your nose in place of another tree that hopes to be fertilized and inhale. And if you're 1,000 feet away, you're going to be pretty much isolated from that tree. So it's known what the major sources of pollen are, and these are data from the northeastern part of the United States. And the change in annual production and change in peak value, the biggest source of pollens are the oaks. Ragweeds, oops, surprisingly, ragweeds in red are less substantial, but ragweed pollen is a major problem for a lot of people. Its scientific name is ambrosia. Don't you like that? Trying to make it sound like this is something that you might have for dessert. And it's a major cause of allergic rhinitis and a precipitator of asthma and other respiratory disorders. And due to the effects of climate changes and increases in temperature, there is almost certain to be an extension of the growing season for ragweed that will occur as a result of these two factors working together. And these are data that have been acquired under controlled laboratory conditions where they measure pollen production and the like. And they have also taken ragweed plants and grown them in atmospheres where you can control the partial pressure of carbon dioxide in the air that surrounds them. And they found the increases in height and weight of the plant and the number, length, and weight of flower clusters. So those climate change and increases in atmospheric CO2 are both independent factors that are almost certain to lead to increases in the amount of pollen that comes from ragweed. And the changes in ragweed are almost certain to dwarf any changes in exposures to pollen that are likely to occur as a result of Darling 58 trees. Let me speed this up a little bit here. This is an important graph that shows atmospheric concentration of CO2 as a function of time. And it has an upward trajectory that's inexorable, but if you look at the fine details, you see that every year there is a dip, and this is due to trees, mainly in boreal forests in the northern hemisphere, taking up carbon dioxide as they grow new plants and leaves. And this is a cross-section of a chestnut that was cut down. It's about 10 years old, and one of the things besides the canker, which is over here, I want you to look at the tree ring, the growth rings in this tree. They're very widely spaced. So this tree is a champ in terms of rapid growth. And rapid growth of trees means it's going to take up a lot of carbon dioxide. Now, if you go over to parking area 7 and the newly planted areas behind the garages there, besides getting a nice view of the PV array, and if you peek, you can see 3 electric car charging stations. There are trees that have various abilities to produce a net uptake of CO2 in an urban forest as opposed to a boreal forest. An urban forest is a forest that requires a lot of attention. You know, if you remember what Heiser entrance looked like when they were planting that. There were 6 or 8 trucks out there, cement mixers, guys running around. I didn't see any women running around there. Working with devices that all burned fossil fuels, fertilizers, all of the effort that goes into maintaining an urban forest that produces carbon dioxide. You have to subtract that from the CO2 that is sequestered by the tree. This guy by the name of Dave Novak, who's a retired forestry department employee, has created a table where he ranks trees according to their last positive point. Bigger is better. So the champ on his list is the sweet gum tree, which grows here, tulip trees, white oaks, and there are 3 of them there, are next in line, and I expect that the American chestnut would probably be in this area here. It's a rapid grower like the tulip tree. The river birch, bringing up the rear, has a last positive point of only 60. So in this one location here, you can see examples of different trees and their ability to fixate carbon dioxide. So it seems likely that based on the comments and the information that's been published by the feds, that the petition is going to be granted. And this is based on the draft environmental impact statement and the Animal and Plant Health Inspection Service report. They say that non-regulated status is preferred under a host of federal regulations because the agency has, quote, determined that the Darling 58 American chestnut is unlikely to pose a plant pest risk. And here's some more quotes from the USDA comments. In the long run, it will have positive impacts on increasing biodiversity of animals and microorganisms while decreasing the abundance of some tree species such as oaks. Oaks took over when chestnuts left. If chestnuts return, oaks are probably going to decrease. They're in trouble anyway, so it'd be good to have a good viable tree to take their place. They also say that transgenic chestnuts are not nutritionally different from their wild-type relatives, and that no effects on threatened or endangered species are expected from the gene that metabolizes oxalic acid. And they also say that wheat has no known safety concern for consumption by humans or livestock. There are no expected impacts on diseases, pests, or other organisms. It's unlikely to spread rapidly and behave as a weed or weed risk potential with other plants with which it may interbreed. Now, they point out elsewhere that it's not fair to call this tree a weed. It was there first. Other things that displaced it have weeds. And apart from chestnut blight tolerance, no differences between modified and unmodified trees have been observed in response to plant disease, plant arthropod, or plant environment interactions. So that's a pretty clean bill of health that the federal agencies have given the Darling 58. Now, if we're fortunate enough to get them, it's likely that we'll get between 2 and 5, and the plan is to plant them up on Wildflower Hill, here where these circles are here, 1, 2, 3, 4, 5, if we're lucky. And we anticipate putting a nice sign up there and a bench where you can sit there and ogle the tree. These are where the hybrid chestnuts are that are already in our collection. And in time, we should expect to have fabulous trees at Kendall. Trees that were once a keystone species in eastern forests. And a recognition by ESF that we have many of the resources need to raise Darling 58 trees, and will be a feather in the cap of the Arboretum in Kendall, and provide yet another unique aspect to the Kendall at Oberlin existence. It will enhance our educational programs and an opportunity for additional scientific programs, perhaps in collaboration with Oberlin College and Conservatory along the lines that brought Valerie Trouet to our campus last summer. So this is what we might see 100 years from now. Not we, us, our great-grandchildren. A giant chestnut tree, maybe with a village smithy standing underneath. Who knows, by then there may be a blacksmith working group or a club formed. There's the chairperson right under there. And it looks like a woman to me. And the place, I went to a place and did a bread baking weekend tutorial. And most of the people who were learning blacksmithing were women there. And eventually, maybe we'll have chestnuts roasting on an open fire. So thanks again for coming, and be happy to try to answer any questions.